Epigenetics is the study of changes in organisms caused by modification of gene expression rather than an alteration of the genetic code itself. It transforms thinking about genomes. In addition to changes in the genetic sequence and other influences, this affects how cells are read. It influences age, environment, lifestyle, disease, and risks of cancer. The genome affects genetic change at any stage of life, not just a baby or a grown adult. Prenatal and postnatal environment exposure can affect risk for chronic disease in adulthood. For example, a mother's exposure to pollution can affect the child's susceptibility to asthma. A mother's vitamin D intake during pregnancy can change the DNA methylation that interferes the functioning abilities of the placenta. Studies also have shown that the father's diet can influence the child's mental fitness. One of the many influences in epigenetics is the environmental influence. Pollution can play a huge role in our genes and how we process pollution. Air pollution can alter methyl tags on DNA and increase risk for neurodegenerative disease, for example. But B vitamins can protect against harmful epigenetic effects of pollution. This simply means that even though we are predisposed to different factors that are affected in the world around us, we can use food and vitamins to protect ourselves against this. Different types of exposure that can affect our genes are heavy metals, pesticides, diesel exhaust, tobacco smoke, polycyclic aromatic hydrocarbons, which are found in substances like coal and tar, hormones, radioactivity, viruses, and also bacteria. One of the most interesting influences that I find is the dietary influence. Certain compounds found in food can protect against cancer by adjusting the methyl sites on oncogenes or genes that suppress tumors. Diet can also positively affect the epigenome health, resulting in a healthier individual for the future. For example, a high-fat, low-carb diet can open up chromatin and can cause differentiation in skin, liver, and brain cells. The diet is very powerful and can literally turn on and off genes that affect who we are and how our health history will look. Our dietary influences are also linked to affecting cancer, cognitive function or dysfunction, respiratory, cardiovascular, reproductive, autoimmune, and neurobehavioral illnesses. So what is chromatin and how does it work? Chromatin is the complex of proteins or histones and DNA that is tightly bundled to fit into the nucleus. It can be changed through a variety of substances like acetyl groups, enzymes, and forms of RNA and microRNA. Tightly folded chromatin tends to be turned off or not expressed, while others can tend to stay open and be expressed as functional. The histones are packaged DNA into chromatin inside a cell. So how does this affect our diet? The influence can be affected through mental ability through the HDAC receivers, for example. A high-fat, low-carb diet is a good example of this. Through our diets and what we choose to eat, chromatin can either be exclusively released or inexclusively released. The amount that's released is up to you and your body and what you decide to eat. Here's the importance of gene expression. Gene expression is divided into two categories, active and inactive. An important process in gene expression is DNA methylation because it controls gene silencing or addition or removal of a methyl group. One of the more popular ways that people have been able to discover and understand gene expression was found in human cancer cells, but later then discovered onto other illnesses and health conditions. It's directly correlated to long-term memory function, chromatin remodeling, and histone modification or gene silencing. These can all affect cancers, mental retardation-associated disorders, immune disorders, neuropsychiatric disorders, and pediatric disorders. Simply speaking, gene expression can be heavily influenced on what we eat and whether or not we're susceptible to diseases and neurological disorders. The chemistry associated with gene activation or inactivation helps create developmental pathways, respond to environmental stimuli, and adapt to new food sources. Genes can control what we eat, how we eat, and different factors that influence our lives. Here's the science between gene activation. Any step of the process can be modulated. Transcriptional initiation, RNA processing, or post-translational modification included. One gene regulator controls the next. So here's what it does. Gene regulation controls cellular differentiation and morphogenesis, or how the embryo is shaped. And it leads to different types of cells that create unique genome sequences, which is why every person in the world is different. This is also why evolution helps us adapt to new things and develop evolutionarily. Here's how it works. These are the stages where gene expression occurs. The most prevalent point is transcription initiation, and this is where chromatin demands. Proteins are also required to bind different elements of DNA or start the transcription process. DNA is read and complementary RNA strand is produced. Different RNA strands are taken out because they are not of use. Transcription then controls the DNA shape and how much RNA is created. DNA is dominated by its structure, and the density is directly resulted from the frequency of transcription. So depending on how much DNA is going through and how frequently it goes through will give you the outcome. And this is also how epigenetics plays a role in what foods we eat. Our environmental factors can all affect how this is completed. This process can also be modified through phosphorylation or methylation and can lead to permanent changes in our gene expression level. Chemically speaking, methylation of DNA is a form of gene silencing. Different enzymes can affect this process. Histone acetylation enzymes are disassociated from their DNA proteins, which allow transcription to occur. This is how the whole process gets started. Post-transcriptional modification is after the DNA is transcribed, the mRNA is formed and translated into proteins. This is completed through capping and splicing of the polytails, much like how we learned in class. Then completed is the RNA transport. This is what moves RNA from the transcription site to the nuclear pore and then to the cytoplasm for translation. mRNA degradation is the mature mRNA that is exported to complete gene expression and create amino acids to form proteins. Proteins are then utilized where necessary in the body. Epigenetics can affect this all because every cell is unique and every gene is differently used in the body due to where it's most prevalent. Here's how gene expression works. It can create proteins, RNA, or tRNA, and membranes are made into cells. This is completed in the cytoplasm, and DNA is found in the nucleus. So first we have our strand of DNA. A complementary strand of RNA is then created. A protein will come in and break down the RNA that is needed, kind of like a little Pac-Man. And then whatever DNA or RNA is not used as the protein goes through will be spit out in a way, and the not useful RNA then exits the nucleus. The strand that made it through then goes through translation. In translation, the strand of our RNA that made it on will go through and enter through these ribosomes here, and when it comes out on the other side, you have a whole unique strand that comes out. That strand is then tightly wound up into a protein. The protein then will get used and built up by amino acids and used in the body for whatever function it needs to be used for. Here's what research has shown in the relationship between epigenetics and the environment. We can learn how to control the embryos, turn on and off genes, and see what factors can affect the embryo. This can teach us what we know about diseases, cancers, and autoimmune disorders and other health conditions, for example. By what we eat and how we live our lives and what kind of environment we'll surround ourselves in, this all affects our genes and who we are. This is why every person is found to be unique and not one person is like any other. Epigenetics has really been a huge factor in science and how we can understand and learn different types of health conditions and health behaviors and diseases that are found in people. We can also learn how the environment can affect who we are and what kind of diseases that we can be predisposed to. Epigenetics is the future of studying and improving overall health care and how we can become a healthier population. Chromatin precipitation, our future of science. This is used to determine whether or not a protein will bind or to its specific site or to a certain DNA sequence. This can also affect how our body reacts to environmental triggers, sickness, and health factors. How it works is DNA binding proteins are linked between DNA to form a formaldehyde in vivo. The chromatin is then isolated and can shear the DNA with bound proteins into small fragments. Certain antibodies that are specific to DNA binding proteins can then be isolated through precipitation. This reverses the crosslinks that release the DNA and digest the proteins. This is then used to specific DNA sequences to see if they were precipitated with the antibody.